All right, good afternoon, everyone. Welcome to the first in our spring series on produce safety. This is webinar number 1. Towards the end, I will share about the other 2 upcoming webinars that we have planned in this series. We've been doing this for several years. Really appreciate the opportunity to engage with our audiences, help to improve food safety practices on farm, help you with some of your knowledge areas, and hopefully implement safer food practices and ACER inspections. So I'm going to go ahead and get started here. Today's webinar is developing environmental monitoring programs, and we have a very special guest presenter with us, Dr. Claire Murphy, who's a food safety specialist at Washington State University, and she's located in Prosser. And we'll give her a little bit more introduction here in a few minutes. Just a few general housekeeping tips to help the webinar run smoothly for you. It does help if you're having trouble with audio or video. It might be our side, but more than likely it's your side and its internet connections, especially if you're in a rural area or you're having inclement weather. We're definitely having some wind and rain today here in the Boise area. It helps if you close all your other programs that are running on your computer. Check your sound. You might make sure that you've got the correct microphone on. And then also you might, if you really have problems with sound, you can always call in and listen to the audio on your phone if your computer's not working. You are all muted. You can't speak during the webinar. I mean, you can speak, just not to us. But if you do have questions for us, you can type them into the chat or the Q&A box, and we will be addressing those as they come in. And I just realized that I've got a typo on this slide. We'll be recording this and our other webinars, and we will have those posted, not last year, but this year in 2024. And you will receive an email with that link. So we have a few goals for today. We're hoping that after participating in this webinar, you will learn more about some of the risks to produce safety. We hope that you will increase your knowledge of how to address risk factors present in your operation through environmental monitoring, and be more prepared to perhaps develop an environmental monitoring program, either in your own operation or in your outreach work. So I will say that all of these webinars were designed in order to help increase the knowledge and skills of our growers, and the topics that we cover come from two places. One are regulators at Idaho State Department of Agriculture. They give me information on where they have noticed compliance issues or issues when they've been doing inspections. I don't get any particular knowledge. I don't know who you are out there, but I do find out that, we see a number of issues related to record keeping or post-harvest sanitation or employee training. And so we use that information to help craft a webinar series that is going to address some of the most pressing issues that we're seeing out in the field. The other source of information for these trainings is if you've been through one of our Produce Safety Alliance grower trainings, you will know that in the evaluation, you have a number of opportunities to share with us areas in which you feel like you need more training, or you still have questions, or you're having issues. And so we will fold those in as well into our webinar topics. So we really hope that you like this one. And this webinar in particular is an opportunity for us to introduce a brand new partner to the Pacific Northwest Regional Food Safety Team. She has not been in her position very long at all. And so we were very excited to be able to bring her on to introduce herself as an expert and a resource in our area, and also to share with us her work in environmental monitoring. So Claire is a food safety specialist with Washington State University, located in Prosser. She joined the WSU School of Food Science as an assistant professor and extension specialist in produce safety in August of 2023, and she's located at the Irrigated Agriculture Research and Extension Center in Prosser. And her program focuses on reducing foodborne pathogen contamination throughout the produce supply chain through integrated research and extension activities. And so we are really happy that her extension activities extend beyond the Washington border and allow her to come in and talk to our Idaho growers, because we don't have a produce safety extension specialist in Idaho. And so we like to borrow from our surrounding states. So that's all I have to get us started. I will come on again at the end of the webinar with some information about what's coming next and also just a wrap up and an evaluation. So thank you, Claire. I am going to stop my share and allow you to bring up yours, and the time is yours. We will try to get through. So we've got about 5 minutes of questions. I really only need probably 5 minutes for my wrap up slides when we get there.
Okay, perfect. Thanks so much for that introduction. Let me make sure it looks good on my end. If you're having any issues hearing or seeing anything, just let me know. Well, thanks so much for inviting me to be here. I'm really excited to get to talk to you all today about environmental monitoring. It's something that I have some research that's starting. Also, it's been something that I know here in Washington, there's been an interest in learning more about. But like Ariel said, I am in Washington, but I'm here in the Pacific Northwest to help with produce safety across the region. So I hope that now that I'm here, I can be a resource to you all as well. So before we get started on environmental monitoring, I just did want to give a little bit of background about myself and my path to Washington State University. So I am originally from the East Coast. I grew up a few different areas there, but have spent the majority of my early years in the East Coast. I ended up at the University of Delaware for the beginning of my academic career. And then I moved to Virginia Tech, where I spent a lot of time. I ended up getting 3 graduate degrees. So I got a master's in food science and got to spend time here on the eastern shore of Virginia, where a lot of tomato production was. And then I moved to main campus. So I spent time on a research station and then moved to main campus where I did a master's of public health and infectious diseases, and then a PhD in food science where I focused all of that on agricultural water. So really looking forward to the new role coming out and to get to apply some of the things I learned there. And then I moved here to Washington State University in August of 2023, and I started my role as an assistant professor and extension specialist, and I do 30% research and 70% extension. And so I am located at the Washington State University Irrigated Agriculture Research and Extension Center in Prosser. So if you ever find yourself out this way, I am located in this big building right here, but I also have a biosafety level 2 lab right over here. But it's a really great facility where both Washington State University, we have a USDA ARS, as well as some Washington State Department of Ag researchers who are all really looking at irrigated agriculture and helping to sustain and improve production. And it's a really large facility. We have a lot of really great resources here. So I have a biosafety level 2, but we can also see we have lots of field trial blocks as well as greenhouses. So there's a lot that is going on here, and it's a really exciting place to get to do research. So my research program has 3 main objectives that I like to break it out into. So my goal is to understand the ecology and epidemiology of foodborne pathogens. So I want to understand where our hazards are. Where are we finding hazards in produce environments so that we can move on to the next one, which is strategies to reduce that risk. So once we know where we're finding pathogens, we can investigate how to best reduce the risk of cross-contamination. And then I like to pull in my master's of public health and do a lot of predictive modeling. We have a lot of data available to us as in this agricultural space. So let's use it to help us make decisions and do some prediction as to where we might find some pathogens. And then of course, my program is 70% extension. And so I find that the two though are really difficult to separate from each other. I think the research itself should support the extension activities like this, talking about research, talking about education, but also the extension should help inform what is next. So based on what I learned from my stakeholders and stakeholders across the Pacific Northwest will help me decide what research needs to be done next. So just that was a quick overview of who I am in my path to Washington State University. But I'm excited to talk to you all today about environmental monitoring. And so I'm going to start us off by thinking about how do we actually go about developing an effective environmental monitoring program. And what this tends to be when I talk to producers is they're in a mindset of checking a box that, yes, I have that environmental monitoring program, but I want to make sure that you have an environmental monitoring program that is useful, that is giving you information that helps you reduce your risk. And so when I start this conversation with producers or extension agents or other auditors, I like to start by understanding like why you want to have an environmental monitoring program. Is it because you want to predict loss of control? You want to understand where those risks are coming in. You want to monitor that environment. Are you using it to evaluate your sanitation? So making sure that if we're doing all of the sanitation work, that it is successful and we're doing it in a way that is actually reducing our environmental risk. Are we identifying weak points in our system where we might have failures in cleaning and sanitizing, or we might need to identify new techniques or approaches for dealing with those weak points? And then also, are we going to build upon existing programming? So why do we want to develop this program? Again, cleaning and sanitation verification. You're going through this action of cleaning and sanitizing, but how well are you actually doing it? We're going to do risk management, so preventing foodborne pathogens from cross-contamination. It might be an investigation. You might have found something within your operation, and now we need to go back and we need to understand how it's getting into our system. I also know that this might be a buyer or an auditor requirement or regulatory requirement, depending on what you fall under. And so if this is a requirement and you have to have an environmental monitoring program, let's make sure we're using it successfully and it's giving us the information that we need. So we're going to start off by understanding when we're developing our environmental monitoring program, what are we looking for? And this is going to be individualized to your individual operation or that operation that you are working with. But what we're looking for is microorganisms. And so we're going to talk through what types of microorganisms we can look for and why we might choose one over the other. So the first one is our indicator organisms. And so these are groups of organisms whose presence reflect the general microbial condition of the environment. So they do help us determine the hygienic status of equipment or environments. They help us validate and verify cleaning and sanitation. They can assess our risk of cross-contamination. They can help us identify some trends that might be going on or identify niches in our environment. So niches where we really need to either readjust our sanitation or we need to focus more on reducing that risk from coming in. What they don't do is they do not indicate the presence or absence of a pathogen. So we cannot use indicator organisms to say whether or not we will have a pathogen in our environment. So typically when we do environmental monitoring for indicator organisms, we're looking at something like an anaerobic plate count. And so this counts all the bacteria that'll grow in the presence of oxygen that have the nutrients available. And so we really see this done a lot for setting a threshold for cleaning and sanitation. So we want our anaerobic plate count, which we often will hear as APC or total plate count. We want it to be at a certain threshold that'll show that we were successful in cleaning. And you'll get to establish that threshold, and we'll talk about how to do that. Coliforms is the same idea. Coliforms is a little bit of a smaller group than anaerobic plate count. It helps us identify some of our routes of fecal contamination. And so this might be something we use if we have concerns related to fecal contamination, which would be something like E. coli, or if we're using a lot of manure in our fields. Yeast and molds, this is to help us with quality. And then we have Enterobacteriaceae. So this is a subgroup of our coliforms. We often see this in dry environments. And so it's a really great way to even get smaller than that coliform testing. And then the last one I'm going to talk about is Listeria species. And so Listeria species is an indicator organism. And this is the one that I'm actually doing some work with here in the state of Washington around developing environmental monitoring for Listeria species. So Listeria species is used as an indicator. And so not all Listeria species are Listeria monocytogenes. So when we test for Listeria species, we're testing for a bunch of different types of Listeria. So we can see here that Listeria includes not all of these. There's 21 currently different Listeria species, but we have Inocua, we have Welsh Mary, we have Celegree. So it's testing for more than just Listeria monocytogenes. And it lets us understand if we have an environment that might support Listeria monocytogenes, which is one of our biggest concerns when we're thinking about the post-harvest environment for produce. So in 2017, the FDA did release a guidance document, and it was called Listeria monocytogenes in ready-to-eat foods, and this did apply also to produce packing houses, and it did focus on testing zone 1s for Listeria species as an indicator organism. And so again, a positive for Listeria species does not indicate that you have the presence of Listeria monocytogenes. And so a few years ago, the predecessor in my role did some work with Listeria species in apple packing houses, and they tested zone 1s, and it was a really useful project because they found that they were finding Listeria species a lot, or the highest place they were finding it was on brush rollers on the wax beds. And so it helped us really understand for the apple industry where we need to focus some of our efforts for reducing that cross-contamination risk. Because if we have that supportive Listeria species, it shows that it could support Listeria monocytogenes. So this is one that I know people are often really afraid of, but I think it's a great thing we can use in the industry to understand where our highest risks might be. So the next thing you might consider testing for is foodborne pathogens. And so when we're talking about the produce industry, Listeria monocytogenes, Salmonella, and then our sugar toxin producing E. coli are the ones we might focus on. I do want to point out that regardless of what you pick, a big misconception that I see when people are getting started developing their environmental monitoring program or changing how they're running their environmental monitoring program is that they see either a positive, so if you get a positive Listeria species, or if you get counts of your total coliforms that exceed the limit that you set, that this is a failure, but it's not. That means your program is working. So the point of having a program is for us to understand where we have some failures or where we need to focus in our operation. And so if you aren't finding it, I'm saying that you're probably not looking in the right environment for it. And so I know that when we get a positive or when we get a limit that exceeds what we set as our threshold, this might be scary. But it's a good thing because it helps us to stop an issue from spreading to be an even larger issue. So I want you and your facilities or whoever you're working with to be prepared to find what you're looking for, because that's the goal. It helps us understand more and helps us continue to reduce that risk. And so why we really want to find it is because we want to stop populations becoming resident. So we want to find populations while they are still transient. So once bacteria have been introduced into the processing environment, we often find them to be one of these two things. They're either resident or transient. So transient don't establish themselves permanently. So it's like our bacteria on the skateboard. They come in and they go right out. And so we typically find that they're being introduced to the facility through raw materials, personnel, maybe pests, but that we have a program in place to find them and to get rid of them so that they just move along. What we're concerned about is pathogens that establish themselves. So they establish themselves in harborage sites, and then they start to multiply, they start to form biofilms, and they persist for a really long period of time. It could be years, it could be months. And so we want to stop them, find them when they're transient, and stop them from becoming resident, because it's a lot more difficult to get bacteria out once they've established themselves. And once they've established themselves, then they start to move around within your facility. And so they might have established in a biofilm in a drain, but then we walk across that drain. We drive our forklift across that drain, and now we're moving it around, and it's making it even more difficult to get it out of the facility. The last thing you might test for, and we don't see it a lot in the produce industry, but it's still something that we do develop environmental monitoring programs for, is allergens. And so there's a lot of different types of swab methods out there if you're concerned about a specific allergen. Okay, so once you've decided I'm gonna test for a certain type of microorganism within my facility, we need to decide where we're going to test for them. So where are we going to look for them when we're doing our environmental monitoring? So the most important thing is to understand, when does that hazard become a risk? And so the hazard itself is the biological, chemical, or physical agent that has that potential to cause illness or injury. So in this case, we're thinking about microorganisms, so biological agents. If you are doing a allergen, that would be chemical. But again, where do we see this hazard? And so I always like to give an example of like hazards versus risks, because I find that these words are often used interchangeably, but they're different. So the hazard in this photo I have is the shark. So the shark itself is the hazard, but we have our swimmer, he's on land, it's not really a risk to him. So once he gets into the water and he starts swimming with the shark is when we see that risk. So that risk is the probability of that hazard not only occurring, but causing serious or adverse health effects. So when we talk about this in terms of microbiological agents, I like to show this type of matrix. So what is the severity of that hazard? So how severe would the outcome be if our product had Listeria monocytogenes or total coliforms? And then what is the likelihood? So if you have low severity and low likelihood, you have pretty low risk. But as we move up to high severity and high likelihood, now our risk level is much higher. So when we're thinking about where we're going to sample, we want to think about how our product moves throughout the operation. So the product flow, when do we see hazards becoming risks? And this can be looking at historical data. So this could be historical data from your operation if you have it. So where have we found samples before? This could be looking at historical outbreaks. So if we know that our product has been associated with outbreaks of certain pathogens and it's been traced back to certain types of environments, so post-harvest water or wax brushes. So we haven't seen that, but that's where the historical data in research has shown us that we're finding it. And so that's a really great place to start if you have that historical data from outbreaks, recalls, or research done by academic institutions. And then proximity is really important, and that's what we're going to move into next. So proximity to our product. So we have our different zones in our packing house. So our zone 1 is what I mentioned already, but that's our food contact surfaces. So this is what is going to come in direct contact with our produce. So it poses the highest risk of contamination because anything that is on that food contact surface is coming in direct contact with our produce. So we can see that pathway of risk, that transfer. And so in zone 1, if we're going to test in zone 1s, we're often doing our ATPs. So those are those different types of indicator organisms for cleanliness, sanitation. We have our indicators, so APC, anaerobic plate count, total coliforms, as well as Listeria species. But we're rarely going to test for pathogens on our zone 1. And this is because, of course, for things like Listeria monocytogenes, we have a 0 tolerance. So if we find it in zone 1, we have to initiate a recall. And just because, again, we find it doesn't mean it necessarily has presented that risk of contamination quite yet. So that's why we often don't see zone 1s testing for pathogens. As we move up to zone 2s, these are our non-food contact surfaces that are close to our food contact surfaces. So we often see this as like sides of equipment. So thinking about what might be close to those zone ones, what might have that risk of venturing into our food contact surfaces. And here's when we're going to start testing for pathogens. So this is where I see operations, and I would recommend testing for Listeria monocytogenes or Salmonella. And then in zone 3s and 4s, so these are adjacent to our non-food contact surfaces. These are things like the floors, the drains, the forklifts. And then zone 4s are outside of that operation, but are still on the premise. So thinking about our break rooms. And this is where we'll test for pathogens, because we want to stop them as they are adjacent to our contact surfaces, as they are outside of our operation, from getting into that operation. So zone 1, of course, when we think about it, is the highest risk in moving outward. So ultimately, when we're thinking about the zones we're going to sample and where within those zones we want to sample, we want to think about, again, those high risks. So where do we find it historically? And this is often harborage niches. And so what we're really trying to do is we're trying to identify where it's harboring so we can stop it from continuing to be in our operation. So we can see here some examples of harborage niches that have been identified previously as high risk. So we have things like improper welds. So bacteria really love to get into these little niches, and it's very difficult to get them out. So thinking about where in our operation we might have some niches, some welds. We have niches here as well that are really difficult to get in and clean. When we're thinking about produce, a lot of these pathogens really love moist environments. They don't have to be direct food contact surfaces, but it's letting them survive within our environment. So we see them in cold storages and then puddles on the floor, as well as our drains. And so pooling water is something we really want to look out for, and make sure that if we have pooling water, we're getting rid of that, because that's going to become a harborage site for us. And then here I have a picture of a forklift wheel. I think we'll have time. I want to, if so, I'm going to show you some research I did in Virginia, but we found a lot of bacteria on these forklift wheels that were moving that pathogen throughout the environment. So thinking of places that move. So forklift wheels, people's shoes, things that are moving throughout your environment. And so how you're going to sample, there's a few different ways to sample, and it depends again on what you've decided to look for. Regardless, it's important to keep aseptic technique. So we want to think about how we're sampling and making sure that, especially if we're looking for something like ATP or anaerobic plate counts, that are some of that organic material or very non-selective bacteria, because it's anything that could grow at oxygen, that we aren't contaminating it. So we want to wear gloves. We want to make sure we're changing our gloves and really making sure that we aren't contaminating our sample. So if you're doing something like allergens or an ATP, a lot of times there's a really standard way. You'll buy a meter, and it'll have specific swabs that you'll use, and pretty straightforward, easy to use. You'll get pretty instantaneous results. If you're doing a swab for something like an indicator or a pathogen, there's different types of swabs, and it's going to depend really on the surface area. So we have our larger swabs here. This is typically what we'll use for a pathogen. We'll swab it on a much larger surface. And then we have our smaller ones, which we'll use for total coliforms, APC, and we'll use them on a smaller surface. Usually we'll have like a 4x4 area, and it just helps us to understand and keep that consistent if we're looking for specific numbers. It's important to think about the moisture. So moisture is going to really help us recover those bacteria. And so a lot of these swabs might come pre-moistened, or you might need to moisten them yourself, but it's how we're going to help get that bacteria off the surface. When thinking of what you're going to moisten it with, or what type of pre-moistened swab, you might want to think about getting one that has a neutralizing buffer. This is really important for understanding if that cleaning and sanitizing is working. So we want to neutralize any of our residual sanitizers so that they aren't continuing to react as we continue to take that sample. We want to understand the snapshot at that moment in time. Okay, and so once we've done this, we've decided what we're looking for and where we're going to find them. Let's decide how we're going to use this data. So in our monitoring, if you don't already have an EMP, so an environmental monitoring program in place, you're going to want to start by just establishing what we call our baseline data. So this is where you're going to go out and you're going to cast a really large net, and you're going to understand the populations of the microorganisms that you have and that you're targeting for your program, and you're going to really evaluate those common population. So I said that we shouldn't be afraid to find total coliforms, and we're going to use our baseline data to understand what that threshold is. What do we typically see? And so we're going to cast that really large net to determine what we typically see, because you're going to find some. But that'll help us understand what to set as our acceptance and rejection criteria. So something like total coliforms, we're going to decide what our baseline data is. What do we usually see, so that if we have elevated values, we know where to set that rejection criteria. For presence absence of like a foodborne pathogen or an indicator like Listeria species, that's where we're going to set up some process control validation. So any presumptive positive is going to be considered a rejection criteria. And then we can start root cause analysis from there. And so we can use the data, whether it's from positive presence absence or accept reject, to start to understand what went wrong and do that root cause analysis to understand how we can prevent that deviation from happening again. So if it's a presence absence, we're going to do some vector swabbing. So I have a square, a rectangle here, and the star right here is where we found a positive Listeria species sample on our brush bed. So we are going to take additional samples around that positive. So we're going to vector swab. So we're going to go take 5 to 15 more to understand how far our contamination has spread. We want to understand how widespread it is, so we can really hone in on what the problem is and determine a solution. So if this was just one sporadic event versus do we have a contamination across our entire brush bed? It's really important, if we're doing vector swabbing, that we're not pooling the data. So we're not going to take all 15 and we're going to put them together and process them at once, because then we don't understand how far that pooling has gone. So some corrective actions that we can do, regardless of what you're testing for. So we want to set corrective actions. They should be predetermined before you've gone out and done your environmental monitoring, so you know what to do once you find that deviation or that rejection. So in our zone 1, we can think about vector swabbing if we're doing presence absence, or if we have a really high elevated total coliforms in one area, let's see how far out that's going. We can break down the equipment if possible and re-clean and sanitize. If we're doing vector swabbing and we're going to re-clean and sanitize, we want to go back and re-swab, and we're going to look for 3 consecutive either negative results or acceptance results to say that we have solved our problem. So that's usually typically what I look for, is did you solve it with 3 consecutive environmental monitorings? In zone 2 and 3, we're going to take a very similar approach to zone 1. And then in zone 4, if we find it, this is our least risky zone. So we're going to start to limit access. We're going to understand where it's coming in so that we can stop it from getting into our zone 3. So I do have some time. So I'm going to talk about a project that I did back in Virginia that really followed this path. So what we did is, a few years ago, I did a project where we went to a lot of different packing houses across the East Coast, and we found one in Virginia that had a lot of Listeria species when we were testing zone 1. And so they allowed us to come back and do a pretty intensive swabbing with them to kind of understand where their environmental monitoring program was failing and also where we were finding that Listeria species. So we went into this tree fruit packing house in Virginia. We went 3 times over a packing season, and we collected 402 samples in total. And each visit, we collected samples before operation and then after operation. And we went to 6 different categories. So we focused here on zones 2, 3, and 4 because we wanted to do a little bit more investigating into the actual pathogen. So no zone 1s on this one. So we looked at cold storage, dry non-food contact surfaces, drains, forklifts, outside of the packing area, so our zone 4s, and then our wet non-food contact surfaces. So what we found was that 35.8% of our results of our 402 swabs were positive for Listeria species, and 24.4 were positive for Listeria monocytogenes. Interestingly, we found no difference between sampling visits and operational time points, operational time points pre and post for both Listeria species and for Listeria monocytogenes. And so if we're thinking about that pre and post, so at the beginning of the day, they should have already cleaned and sanitized from the day before. The end of the day, we would expect that the line has that buildup of organic matter. It might have that higher prevalence. So with that, because we weren't seeing that difference in pre and post, that showed us that the frequency and thoroughness of their cleaning wasn't efficient at reducing that risk and controlling that Listeria monocytogenes or that Listeria species, which is another way, again, we can use our environmental monitoring program before and after something like cleaning to understand if it's doing its job. So no significant difference, so it wasn't doing its job. We did find a significant difference between both Listeria monocytogenes and Listeria species in the zones, and we found that zone 3 had substantially higher Listeria monocytogenes and species. So it was getting into our environment, but we weren't seeing it move as quickly through. So we looked for mono in all of our species, and we found, of course, mono of all the species was the most prevalent, followed by Celery, Inocua, Inovii, Marthi, and Welshberry. So these are all some of those non-pathogenic indicators. Interestingly, we also found that for 30 of the positive sites, we had two species present. So we were getting Listeria species and Listeria inocua. So that shows that if we're getting something like inocua or Marthi, it does truly indicate that we might have that presence. So something really interesting that we can lean back to say, yes, if we're testing for species, it is going to have us have that good understanding of the presence in that environment. Does it support? Of course, again, does not mean it's always present. So of our 6 types of categories, we found that Listeria and Listeria monocytogenes were most present in our cold storage, our drains, in our wet food contact surfaces. So thinking back to a few slides ago when we talked about those harborage sites, we're finding them in those wet environments, those moist environments. And this packing house had a lot of issues with maintaining the moisture within their operation. So that made sense to us. They had a lot of moisture buildup. They had a lot of pooling water. And so we took this one step further, which what I think is really interesting is we did some sequencing. So not typically done at an institution. We have a lot of these resources available. And because we saw monocytogenes to be the most prevalent, we focused on 3 different allelotypes. So these are fingerprints, basically. So these 3 different Listeria monocytogenes are from the same family, so the same fingerprint. So we wanted to watch them move throughout the environment because we found the 57 19 times, the 62 13 times, and the 112 11 times. So we're seeing that these 3 have maybe become resident. So we're moving out of transient and we're moving into resident. And so what this figure is showing us, and I'm just going to show us one because again, I just wanted to give a really big overview of like what we can learn from environmental monitoring, is the different colors here are the different visits. So we went 3 times, and then the different letters or that actual fingerprint. So we saw that we had the fingerprint that we labeled B in our hydro cooler. It was on our forklift on the same visit, but on a different visit, we have it moving to our packing line. So because we started to have this transient listeria, we start to see it moving. And we see that here with C as well. So it was on our forklift on one visit and our bins, but then it's moving again. So thinking about why we really want to stop these problems, why we want to identify it, why it's really important to identify where we're having these elevated pathogen indicators, or where we're having elevated coliforms, because it'll help us identify if we're finding it in our hydro cooler. Think about all that traffic that goes through the hydro cooler, and we clearly were seeing that resident population moving. So really interesting to me to see how we found it, and because we had that inefficient sanitation program in this operation, it was moving. So some final thoughts is talking about not only the environmental monitoring program, but all that we have talked about over the last 30 minutes is we want to use environmental monitoring programs as an early warning sign for hazards. So predicting where they are before they become that risk, before we lose that control in our operation. Swabbing for indicator organisms can be really useful at validating and verifying that sanitation program. So taking those swabs before and after and seeing that change. Focusing that on hard to reach, those niches, those places that we don't think of as cleaning, so we can understand where they might be and where they're spreading from. Again, this is going to be really individualized to your operation. It's going to be based on the historical data in your operation, historical data related to that produce, what you think your biggest hazards and risks are. You're going to find these organisms, and that's okay. That's what I want you to do. If you weren't finding them, then I'd say we need to reevaluate your environmental monitoring program. Let's reevaluate what we're testing for, where we're testing, how we're testing. And then having those corrective actions in place, because we're going to find them, and we want to know what we want to do when we have those elevated risks, because that'll help us know what to do in the moment and understand how to best react when we do see those breaks in protocol. So that's all I had, but I know we still have a decent amount of time, and I see a few chats, and so I have my email up there. Of course, I'm really here to be a resource here in the Pacific Northwest, but I also want to open up for any questions related to environmental monitoring or any other topics related to produce safety that I can touch upon.
Thank you so much, Claire. Those were really, that was really a fabulous presentation. I feel like it explained that in a way that made a lot of sense to me as someone who has not implemented environmental monitoring programs.
Yeah.
I have a couple questions while we wait for some more questions to come through in the chat. So your examples showed us some testing and some monitoring in an apple packing houses, and that is definitely relevant to a lot of Idaho growers. What are some other examples of how and when and where one might do some monitoring in, say, a diversified vegetable, like small scale operation?
Yeah, so I think that again, so if you haven't established a baseline, that's a great place to start. So you're going to go in, and so we see, of course, vegetable operations being more keen to some of our E. coli. So I would focus on, I would start with your total coliforms and understand where we're just having some higher risks. So where do we have high levels of total coliforms? Because that's going to be in that same family as our E. coli's, and that's more of what we're thinking about with some of our vegetable production. But it doesn't matter the size of the operation. I think we can learn a lot by going in and taking some of those zone ones in places that are really high touch, but then also thinking about where are the places that maybe we aren't cleaning. And so I think that they all, regardless of the size, is really applicable. And something even as like an ATP meter gives us really quick results. So something like listeria, we might have to wait 48 hours to get that result, but an ATP is a great way to start because it gives us that instantaneous result. It's going to be high because we're just looking for organic material, but organic material, when we think about it, it's that soil, it's that dirt. So where are we having that buildup of debris?
That's a great answer. Now, an ATP monitor, is that something that folks can buy, like at Granger or fairly easily?
Yeah, so they're pretty easy. I'm pretty sure you can get one at Granger. I buy mine from Fisher Scientific, somewhere also that I see a lot of people use. And then you'll just want to buy, and they're usually about $1,200 for the actual meter themselves. And then you'll buy the swabs that go with it. And I usually can buy about 100 swabs for about $50. So not too expensive. And it'll give you a pretty quick result because it is truly just looking for that presence of organic material.
Right. And I know we have some growers, especially like coming back to our vegetable producers that have outdoor, like outdoor packing areas.
Yeah,
What are some major risks you see in those environments, and where would you focus your efforts?
yeah. So under the produce safety rule, your packing environment can be indoors, but it can also be 4 sticks in a roof. Like there is no requirement under the produce safety rule as to what your packing environment looks like. So when we're talking about risk, it's going to look really different depending on that structure itself. So when we work with operations that have more of that outdoor packing, I see the biggest risk coming from pests. So there, I would spend your time first focusing on your pest management. So if you have birds living in the rafters, let's put some spikes up there, and then let's focus on reducing that intrusion because that's where we find it coming from. And then once you have that pest management program in place, I would focus on starting actually outward to inward if we have an outdoor environment, because that's how they're coming in, is through the environment itself.
That's wonderful. Thank you. And I want to encourage those of you that are on still, please, if you have questions, put them in the Q&A or chat. And actually on the call today too, we have one of our presenters who's going to be joining us at the end of the month, Alexa Johnson from Nevada. And did you have a question or comment, Alexa?
Oh, you're muted, Alexa.
Unmute. Sorry.
Thank
So
you.
I was just kind of curious on that last one of those last slides that you showed where you were talking about the fingerprints and how you found it in different areas, and then from one visit to the next, how it was moving. When you identified that there were 2 forklifts that had different fingerprints on them, did they have their forklifts segregated, like in like certain forklifts are only allowed in certain areas? Like you have a forklift for raw goods versus a forklift for finished goods, and maybe as a potential for that being another way to mitigate how it can be traveling through your facility.
So they did not.
Okay.
And so after we did this research, we spent a lot of time working with that individual packing house because we saw a lot of those issues. And so talking about that segregation, absolutely.
Yeah.
And we talk about that in the produce safety rule with like thinking about managing if you have manure or you have livestock, having separate like PPE, so separate gloves, separate boots for managing livestock in manure before you walk into your production environment or your packing house. But it's the same thing in a packing house or an open pack shed, having that segregated equipment. So using some color coding is a great way to do that because no, they did not have designated forklifts for certain tasks. So they were taking those 4 forklifts all around that operation.
So basically from their dirtiest areas into their cleanest areas and back and forth. Yeah. So, I don't know. It's just kind of interesting when you see how they move, like what different traffic patterns are contributing to that.
Yeah, so we saw a lot of that traffic pattern in that specific one coming from cold rooms. So they had a lot of moisture buildup in their cold rooms. And then the forklifts, we think, were what was moving it to the packing line.
Okay, thanks.
And I have another question for operations that maybe do not have, maybe they do have cold storage, but they are more focused on the vegetables. Would you still want to test cold storage and drains for listeria in addition to looking for E. coli and other coliform?
Yeah, I think it depends on, so I know, and that we as producers are managing lots of different aims, and food safety costs money. And so I know that I could suggest that you test for everything in every environment, but that's not realistic. So going back to like, what is our highest risk? What do we identify as historically associated? We know we find Listeria in drains. That is not something that we're still thinking about. We find Listeria in drains. And so if you don't have the mechanisms to test for that and you want to focus on other things first, just keeping in mind that your drains are going to be a risk, and just thinking about how often are you even approaching your drains as a sanitation? How often are you making sure that they aren't backing up? Do you have backflow prevention on your drains? And so those are things that, like, if you don't have the resources for, I would focus on still those mitigation measures, but focusing your actual environmental monitoring on what we don't understand about your facility, so that we can reduce and identify those risks that are unique to you.
That's great. And I have one more question. So I'm just trying to think about the spectrum of our producers in Idaho. And so we do have a lot of tree fruit. We have a lot of, row crop and diversified vegetable production, but we also have a great deal of onion production. And I know that onions aren't necessarily your specialty, but just thinking about a dry pack house, right, where there is no post-harvest water being used, what would be some of the primary pathogenic risks in an onion packing shed, and what might be tested for in that situation?
Yeah, so historically we've seen salmonella being what we find with our onions. And so Enterobacteriaceae is what we typically use as the good indicator for understanding our dry environments. And so that is one that's going to be a little more on the lines of like total coliforms or Listeria species. So it isn't going to be as quick as a ATP. It's going to require some level of a plate or some microbial media. But that's what we typically see with the dry environments. But we also know that the dry environments are very different, a different beast, because we're not looking for a lot of those moist environments. And so focusing on where we're seeing buildup of debris is a really great way, especially because we aren't using any type of sanitizer that is moving. So a lot of times in the dry environments, we're seeing ethanol or alcohol-based sanitizers, which aren't going to be as sufficient at removing debris. So focusing on where we're seeing that buildup of debris.
Fantastic. Well, gosh, I want to really open that up and offer anybody the opportunity to continue to ask questions, but I will go ahead and move into my closing slides, I guess, if that's okay. And again, thank you, Claire. That was really, really helpful, and something that we have not covered in our webinar series before.
Yeah.
So I really appreciate that that's going to be part of our library now. And along those lines, I do want to mention that we have 2 more webinars coming up this month. So on April 18th, we have Record Keeping for Produce Safety Simplified. And that will be presented by myself with some assistance from Greg Blahato, who is our Idaho Department of Ag Produce Safety Manager. One of the things, like I said, that we find a lot in inspections and when we do on-farm readiness reviews is just, just some misunderstandings or confusion around record keeping, or just ways that record keeping could be more efficient and maybe better represent the good practices that are actually happening on farms. And so we will go through our University of Idaho Extension record keeping guide, the required records, and just kind of have a friendly discussion about getting our records in order for the next year. And then April 25th, we will have Alexa Johnson, who is with the Nevada State Department of Agriculture, and you can see her here on the screen. It's going to talk about something I'm very excited about, because we are seeing a real explosion of both microgreens and mushrooms. So a lot of indoor culture of food crops in the state, and very little that we know about the food safety of that. And so this is a learning opportunity for me, as well as our growers, our farmers market vendors, et cetera, at really any scale of microgreen and mushroom growers. And looking forward to that presentation. So if you have not yet signed up, you can. If you signed up for this webinar, you will be getting the notifications for the next ones coming up. And then I just wanted to remind you that we have over 13 hours of our recorded webinars going back to 2020. We've done 3 or 4 a year. And I encourage you to go back and look at the whole playlist. If there's something in particular that you are struggling with, whether it's worker training, record keeping, cleaning and sanitation was our highlight last year. The year before that, we worked on avian bird pests and rodent pests. So when Claire was mentioning, managing your pests in an outdoor packing environment, we've got 2 fantastic webinars on that. And those are all located on our YouTube channel, which here is the link here. And we will also include that for you in the follow-up email. So that's really all I had. I wanted to thank you all for your attendance today. I want to thank you for being farmers and for having a commitment to a food safe culture. We really appreciate the extra time and effort that you take to focus on food safety. And we will have a quick 3-minute feedback survey that will launch when this webinar closes. Just helps us to get some information on whether we're meeting our goals for today's presentation and meeting your goals for education. So we will also send out Claire's contact information, if that's okay, in the follow-up email. So if there was something that you particularly wanted to discuss with her but didn't want to share with the whole group, you are welcome to reach out. And we really look forward to seeing you in 2 weeks. And thank you, Alexa, for joining us too today. Have a great rest of your week, everyone. Thank you, Claire.
Thank you. And yes, absolutely. You can share out my contact information. Any questions about any topic related to produce safety, happy to answer.
Wonderful. Thanks again.
Of course, have a great rest of your day.
You too.